The next experiment concerned with photosynthesis is Rubin and Kamen experiments. Now, Rubin and Kamen are the scientists. They did experiments in the organism chlorella. Chlorella is a unicellular non-motile algae. So what they did is they have provided water. Now this water is having a heavy oxygen. So this is a heavy isotope. If you can observe, so usually the mass number should be 16, but here they have opted the oxygen with a molecular weight, that is mass number is 18. So this water molecule they have provided to the algae, and along with water they have also provided CO2. And in presence of sunlight, what happened is the algae performed this photosynthesis process, and the products obtained are glucose, water, and oxygen is also released. But the oxygen which is released, O18, with the mass number O18, since they have tagged this mass number O18 to the water, that means what? So if this oxygen is liberated or produced is containing O18 means what? So this oxygen might have released from the water. So this experiment has proved that the oxygen which is released from the or produced by the photosynthesis is actually obtained from the water, not from the CO2. So this is one experiment which proves that the oxygen is released from the water. So the next topic would be the structure of chloroplast. Now we'll go with the structure of chloroplast. As you all aware that in the plant leaves, wherever the green portions are there in the plant, there in those cells they contain an organelle which is a chloroplast, and in this chloroplast chlorophyll pigments are present. Now let's go ahead with the structure of the chloroplast. The chloroplast is a double membrane bound organelle. It has an outer membrane and inner membrane. Thus, it is a double membrane bound organelle. Outer membrane is permeable membrane. It allows all type of molecules inside and outside. Whereas inner membrane is selectively permeable. It allows only selected solutes. Now, if we observe the inside of this chloroplast, there is all liquid proteinaceous material is present. So this all liquid material which is present is stroma. In this stroma, thylakoid membranes are present. Now we can observe the green color membranous layers. So this membranous green color layer is nothing but this is all thylakoid membrane. Right. So this thylakoid membrane is getting folded and forming a piles of coin like structures. These are grana. So this thylakoid membrane folding and forming certain piles of structures which is known as grana. And here we can observe this is a thylakoid membrane. This membrane which is connecting the grana is known as stroma lamellae. Right. Now these thylakoid membrane, whether it is grana or the stroma lamellae or fret membranes, so these are the membranes which contain chlorophyll pigments. ఈ మెంబ్రెయిన్ పైన క్లోరోఫిల్ పిగ్మెంట్స్ అనేవి ఉంటాయి. క్లోరోఫిల్ ఏ, క్లోరోఫిల్ బి అండ్ అదర్ సార్ట్ ఆఫ్ పిగ్మెంట్స్ లైక్ carotenoids, xanthophylls. So all these are the pigments which are present on this thylakoid membrane. And the empty space which we see inside is this region is lumen part. The inner region is lumen part. So any which ways we will learn about this grana and thylakoid the stroma lamellae in the further classes. Now let's focus on the importance of this thylakoid membrane. On the thylakoid membrane, I told you that the chlorophyll pigments are present. So first what happens is, the chlorophyll pigments will be absorbing the light, and the light energy is converted into chemical energy. So on the thylakoid membrane, light energy will be converted into chemical energy. That a detailed process we will be learning in the next classes. So thus the light reactions takes place in the thylakoid membrane. Next, after the light reactions, the assimilatory power ATP and NADPH are produced. The products of light reactions are ATP and NADPH plus H plus. Now this assimilatory power is used for the next sort of reactions, which are dark reactions. Now dark reactions takes place in the stroma. So this is all the stroma. So stroma has various sort of enzymes. So the second step reactions takes place is dark reactions or light independent reactions. So these dark reactions does not takes place in the dark, but it means that these reactions are independent of direct light. వీటికి లైట్ అవసరం లే, కానీ లైట్ ప్రొడక్ట్స్ అవసరం. They doesn't need light, but they need light products. So what happens is, in the stroma, the dark reactions takes place where the first step products ATP, NADPH are used for this dark reaction, and here the chemical energy is converted into food in this dark reaction process, where the chemical energy is converted into glucose. So thus photosynthesis takes place in majorly two stages. The first stage is light reactions, which are also known as photochemical reactions. Light reactions occur on the thylakoid membrane, where the light energy is converted into chemical energy, which is ATP and NADPH plus H plus. And this ATP and NADPH is known as assimilatory power. Now the products of the light reactions are utilized by the dark reactions. Now where does this dark reaction takes place? It takes place in the stroma. So right here what happens is, the assimilatory power ATP, NADPH is used to reduce the carbon, to reduce the CO2, and to produce glucose. So here that chemical energy later converts into food, which is glucose. So that particular stage is dark reactions. And if you can observe the chloroplast, we can also find the circular DNA, we can also find ribosomes, we can find starch granules, and all these things we can able to find in a chloroplast. And chloroplast is known as a semi-autonomous organelle. The next one is, if you can observe, here I have taken two cells, and these are the blue color one dots are nothing but the chloroplast. So here I am representing the chloroplast with a blue color dot. Right. Now here, when there is a low light intensity, now the chloroplasts are arranged perpendicular to the radial wall, because they need light, because it's only low light. So that is why the chloroplasts are arranged parallel to the transverse wall and perpendicular to the radial wall. Itla arrange avutayi enduku, so that they could get proper amount of light for all the chloroplast. The second case, if you can observe this cell here, during high light intensities, the chloroplast inside the cell, they are arranged parallel to the radial walls and perpendicular to the transverse wall. So this is a radial wall, this is a transverse wall. High light intensity appudu chloroplast itla radial wall ki parallel ga arrange ayi untayi, transverse wall ki perpendicular ga arrange ayi untayi, because high light intensity is dangerous for the chloroplast. Okay, so that is why, to escape that high light intensity, they have arranged parallel to these radial walls. So this is about the structure of the chloroplast.